One of the key ways we can try and understand the phenotype of a mutant yeast we've created in the lab is figuring out exactly how the new strain responds to stress. When we're knocking out a gene we hypothesize might be involved in the cellular response to certain kinds of stress, we need a simple, high-throughput assay to see if, in the absence of that gene product, our organism behaves differently than it did before. One of the most important experiments in classical yeast genetics is the spot assay. The concept is relatively simple. We're going to create a serial 10-fold dilution of our mutant strain and spot our dilutions into a solid media containing our stressor of interest. We then compare the growth of our mutant in the presence of that stressor to the background strain, or ancestor. If our knockout grows more poorly under that kind of stress, that means that our gene of interest may have a role in our organism's response to it. This experiment comes in two parts: making a clean, stress-enriched agar surface to spot our cells onto, and diluting the cells to the right concentration to get that characteristic spot assay pattern of growth. First, making the stressor media. Here we go. There are a few things we need before we start mixing. The final volume of solid media in our petri dish is going to be 24 milliliters. We need to be able to mix in enough of our stressor into the plate without compromising the balance of ingredients that make it a nutrient-rich agar. It needs to be identical to a stress-free control, otherwise our conclusions won't be valid. To account for that, we're going to take 12 mils of agar at 2 times the final concentration and keep it just warm enough that it stays molten, but not so hot that it can degrade any of our stressors. Then we're going to take 6 mils of subarachnoid dextrose broth at 4 times the final concentration, as 6 mils is a quarter of our final volume of 24. This gives us 6 mils of water to make up the volume, and this is the part we can modify by adding our stressing agent. I've done some research and found that my gene of interest might have something to do with the integrity of the cell wall. So I'm going to use Congo Red, a bright red compound that can disrupt the cell wall. My stock concentration of Congo Red is at 100 milligrams per milliliter, but that's too toxic and will straight up kill any cells we try and grow on it. So I'm going to want it diluted to 1 milligram per mil in the final plate. That means my 6 mils of water will need to have 4 times the final concentration, 4 milligrams per mil. 100 milligrams per mil stock divided by 4 milligrams per mil final gives us a dilution factor of 25. So we'll need to do a 1 in 25 dilution of Congo Red in our 6 mils of water. 6 mils divided by 25 is 0.24 mils, which means we'll need to take out 240 microliters of water from our 6 mil aliquot and replace it with 240 microliters of Congo Red stock. Now we can mix our stressor solution with our 4 times SD broth and mix that very thoroughly with our warm 2 times agar. Note that we've kept our broth warm as well, because adding cold liquid to agar may cause it to start solidifying, leaving unsightly and annoying chunks in our petri dish. Working as quickly as possible, we then quickly pour our agar mix into a petri dish that we've left open in the biosafety cabinet, trying to work as aseptically as possible to limit the risk of contamination on our carefully handcrafted plate. We want to leave it out for at least 15 minutes until it sets, but leaving it longer is usually better. The drier the plate, the more thirstily it will absorb and dry up your yeast spots, giving you a nice, clean-looking spot assay.